On today's Arabidopsis Research Roundup, I'm joined by Vinod Kumar from the John Innes Centre, and he's talking about a new paper just published in Molecular Plants. And the title of this paper is SWR Chromatin-Remodelling Complex Subunits and Histone 2AZ have non-overlapping functions in immunity and gene regulation in Arabidopsis. So, Vinod, thanks very much for joining us today. So, can you give us a summary of what you've done in the paper?
Absolutely. So as you said, the title says it all. So what we are kind of looking at here is looking at characterizing the roles of the Arabidopsis SWR1 chromatin-remodelling complex in plant immunity. So before that, it will be good to actually put that into a context where this SWR1 complex actually sits in. So this is a big molecular complex which actually involves something like 15 proteins. Okay. So the main role of this complex, as we understand from other organisms, mainly from yeast and things like that, is the incorporation of variant histones. These are histone variants which are evolutionarily conserved but has slight sequence differences from the normal histones that we otherwise find on chromatin. So these kind of gave distinct biochemical and biophysical properties to the nucleosome. So therefore add new dimensions to the regulatory framework. So you have more regulatory innovations that's actually possible. So H2AZ, this is another histone variant. So this has been implicated in roles in plant development and also in response to environmental signals like temperature. Okay. So what we are looking at here, so we actually looked at a battery of mutants, which are mutations in component proteins of the SWR1 complex. So what we started by looking at the immunity phenotypes by challenging these plants with Pseudomonas. So what we are actually looking at is a mutant that is actually in the core enzymatic subunit of the complex. That's 5-1 that corresponds to SWR1. And then two auxiliary subunits which are non-enzymatic. One is acting related protein 6, R6, and SWC6. Okay. And also a mutant which is actually depleted in the H2AZ proteins itself. So what we actually find is that when we actually perturb H2AZ from the chromatin, so it actually affects the ability of the plant to resist itself from a pathogen like Pseudomonas syringae. So, but then when we actually see it, so what we see is that in general, for example, 5-1, SWC6, and the depleted H2AZ line show increased susceptibility, but R6 actually show increased resistance. Okay. So this is unexpected because this is a complex which is meant to have a single function. As we understand from yeast as well, and all the eukaryotic organisms, they are very well conserved. And also in yeast, it actually suggests that R6 and SWC6, these are two proteins which are interdependent, and we are mutually required for each other's presence in the complex and activity. So therefore, you would expect that these two proteins should have same function. Mm-hmm. But when we actually look at the physiological context, they do have different functions. But then if you have to look at the developmental phenotypes of these mutants, they look almost the same. So which means that so it's not all the same, but depending on physiological context or in different pathways, they would have innovative functions and novel functions that we don't actually know at the moment. Okay. So then we went on, we looked at other pathogens, so like virulent pathogens, which actually require the SA-mediated pathway for complete resistance. There again, we actually see that the discrepancies between these mutants exist. So in general, either 5-1 or SWC6 leads to increased susceptibility. R6 remains resistant. Okay. So then what we actually looked at is one of the pathways which is very crucial for resistance to these biotrophic pathogens is accumulation of SA, the salicylic acid. Okay. So when we quantify salicylic, we just asked whether is that because they cannot actually accumulate salicylic acid, which was actually not the case. In fact, these mutants can accumulate or induce accumulation of salicylic acid upon pathogen infection, even much better than the wild type. Therefore, it's not actually failure to accumulate salicylic acid, but actually responding to or the responses downstream to that.
Okay.
So this was very interesting. And then what is more interesting is that, so generally there is that presumed antagonism between SA and JA-mediated pathways to a biotrophic and a necrotrophic pathogen. So generally, if you are compromising resistance to biotropes, you are better off with the necrotrophic pathogen.
Yeah.
So,
Okay.
but when we actually challenge these mutant sets with a necrotrophic pathogen detritus, you still see the same kind of hierarchical results that we find.
Okay.
Suggests that it's much more pleiotropic, and then so we need to actually look at it in a much more careful manner.
Okay.
So then immediately, because H2AZ and these chromatin complexes, so they have their critical roles in gene regulation. We actually looked at the gene expression through RNA-seq. So what we actually find is that we find more differences than similarities between these mutants.
Okay.
So when we actually look at it, so you can do all sorts of analysis. So if we did the principal component analysis to actually see that the mutants, so they actually segregate into 4 different groups where Pi1 and Op6, they are far apart from each other, and then also form a subset of different clade from the other mutant set.
Yeah,
so it suggests that possibly at the molecular level they do have some functional diversity.
Okay.
So when we actually then look at the gene population, so the gene expression phenotypes, you actually see that many of the defense genes are misregulated. You can actually see the basal level of many of the defense genes are up. But then at the same time, what we actually see is it's that very pleiotropic. So you have genes which are required for positive regulation of defense are up as well as the genes that are actually negatively regulating. So then in the end, what the physiological phenotype that we actually see is actually the result of the epistatic interaction between those pathways that are now being perturbed in this mutant. So what it actually says is that one has to be very, very careful in assessing a particular phenotype or the outcome of a mutation, and we have to actually look at multiple readouts to actually determine what the real outcome is of a particular mutation.
Yeah, I see. Yeah. Okay, that's certainly good advice. So do you have an idea that the ARP6 might be functioning in a different way outside of the SWR?
It is very likely that it might have functions outside the SWR1 complex.
Okay.
Or even in addition to that, it is also possible that ARP6 function in the SWR1 complex itself might be slightly different from what we understand from other organisms like in yeast.
Okay.
So it kind of makes sense in plants because being a multicellular organism with such challenging life to deal with, so I think it will not be surprising if you will find molecular innovations in plants for such a fundamental molecular complexes like that of a chromatin-remodelling complex, where the complex components get functionally specialized, or kind of have a specialized functions depending on the developmental or physiological context, that the system can modulate the involvement and function of each component, that will modulate the activity of the complex, and then have a much better or a much efficient regulation of its chromatin.
Yeah. So does the plants which lack the histone 2A-Z component, do they have a additive phenotype, or do they have a defense phenotype at all?
Yes.
Those mutants.
So exactly. So this is what. So we have this line which actually lack histone H2AZ. So they basically mimic the phenotype of the ATPase mutant. That's a Pi1 mutant in a big way. So Pi1, or that's the SWR1 ATPase subunit in the complex. So that should have reduced H2AZ incorporation. So H2AZ mutant actually mimic that the phenotype of Pi1.
Okay.
But then that is not completely overlapping. That's also very surprising to us. Because if you actually look at the gene expression phenotypes, so the correlation between PI1 and the H2S depleted lines are not very impressive. And then it's probably less than what you would expect for something which has such conserved and shared function.
Okay.
So which also suggests that there are possibly PI1 would also have PI1 mutation would also result in other chromatin mediated effect, probably outside of H2AZ. So this we need to be checked out. So this is something that we are actually following up to study what these individual complex components do possess in chromatin remodeling or outside. In the context of the physiological and developmental phenotypes that we see.
Okay, so it's going to be interesting to see in the future whether it's a defense specific phenotypes or whether there's a more general role going on, which I'm sure there probably is, I guess.
Absolutely. So yes, it's really exciting for us because now plants give us a fantastic opportunity to dissect a core molecular complex like that of the SWR1 complex. And then the phenotypes and the molecular outputs that we see indicate that there is much more than what we actually understand of this complex, in Arabidopsis at least.
Cool. All right, so we look forward to seeing that future work. So thanks very much, Vinod, for joining us today. Thanks.
Thank you.